Using mass spectrometry, researchers were able to classify over 400 Alzheimer's patients into 5 distinct subtypes. They accomplished this by analyzing the levels of specific proteins in their cerebrospinal fluid, the clear liquid that surrounds the brain and spinal cord. By studying the variations in protein levels, the researchers were able to identify patterns that could potentially lead to more precise diagnostics and treatment strategies. Each of the 5 subtypes identified by the study corresponds to a distinct pattern of protein levels in the cerebrospinal fluid, reflecting the varying biological processes at play in each individual's condition. This discovery could drastically shift the way Alzheimer's is diagnosed, allowing for more personalized treatment approaches. Instead of a generic approach for all patients, doctors could tailor treatments based on which subtype the patient falls into. Researchers believe that understanding these subtypes might also shed light on the different stages of the disease. Some patients may have a slower progression of symptoms, while others may experience a rapid decline in cognitive function. The differences in protein levels could be linked to these variations, opening up new avenues for monitoring disease progression and intervention. This discovery also highlights the critical role of biomarkers in Alzheimer's research. Biomarkers are measurable substances that can indicate the presence or progression of a disease, and identifying the specific proteins associated with Alzheimer's is a significant step forward. Researchers are now focusing on how these biomarkers can be used to detect Alzheimer's at earlier stages, when treatments might be more effective. Early detection could be the key to preventing or delaying the onset of symptoms, potentially transforming the treatment landscape for the disease. Moreover, this breakthrough also opens up possibilities for new drug development. By targeting the specific proteins linked to each subtype, pharmaceutical companies could create drugs that more precisely address the underlying causes of Alzheimer's in different patient populations. This could lead to treatments that are more effective and less likely to cause side effects, as they would be specifically designed for the biological processes involved in each subtype. While this research is promising, it's important to note that the study is still in its early stages. Researchers need to conduct further studies to confirm the findings and explore the clinical implications of these protein subtypes. However, the potential for this research to revolutionize Alzheimer's diagnosis and treatment is immense, and it's a step forward in understanding a disease that has long been shrouded in mystery. As scientists continue to investigate these findings, they are also exploring how lifestyle factors, such as diet and exercise, may impact the levels of these proteins and potentially influence the development or progression of Alzheimer's. It's becoming increasingly clear that Alzheimer's is not solely a result of aging or genetics, but rather a complex interaction of biological, environmental, and lifestyle factors. This holistic approach to studying the disease could lead to more effective prevention strategies. In conclusion, the use of mass spectrometry to identify distinct subtypes of Alzheimer's disease based on protein levels in cerebrospinal fluid represents a major breakthrough in understanding this devastating condition. It brings us one step closer to more personalized, effective treatments, and may eventually lead to improved outcomes for patients suffering from Alzheimer's. With continued research and innovation, the hope is that these discoveries will pave the way for earlier detection, targeted therapies, and ultimately, a cure for this debilitating disease.